Cells and Microorganisms Complete Learning Guide. 1. Cell structure. All living organisms are made of cells. There are two main types of cells. Eukaryotic cells, found in plants and animals. These cells are larger and more complex. Prokaryotic cells, found in bacteria. These cells are smaller and simpler. Animal cells. Animal cells usually have nucleus, which controls the cell and contains DNA. Cytoplasm, a jelly-like fluid where chemical reactions happen. Cell membrane, which controls what enters and leaves the cell. Mitochondria, which produce energy through respiration. Ribosomes, which make proteins. Plant cells. Plant cells have all of the animal cell parts, plus cell wall, a rigid structure made of cellulose. Permanent vacuole, which contains cell sap. Chloroplasts, which capture sunlight for photosynthesis. Bacterial cells. Bacteria have cytoplasm, a cell membrane, and a cell wall. Their genetic material is a DNA loop in the cytoplasm, and sometimes they have plasmids or small rings of DNA. Suggested images. Labeled diagrams of animal, plant, and bacterial cells side by side. 2. Microscopy. Light microscopes can show basic structures like nucleus, cytoplasm, and chloroplasts. Electron microscopes have higher magnification and can show tiny structures like ribosomes. Practical. Prepare slides, use a microscope, and draw what you see. Key formula. Magnification equals image size divided by real size. Suggested images. Diagram comparing light and electron microscopes with labeled magnification examples. 3. Cell differentiation and specialization. Specialized cells are adapted to perform specific functions. Most animal cells lose the ability to specialize after early development, but many plant cells can specialize throughout their life. Examples of specialized cells: sperm cells, which move to fertilize eggs, nerve cells, which carry electrical signals, muscle cells, which contract to move, root hair cells, which absorb water and minerals, xylem and phloem cells, which transport water and nutrients in plants. Suggested images. Diagrams of specialized cells showing their adaptations. 4. Chromosomes. Body cells contain two sets of chromosomes in the nucleus. Chromosomes are made of DNA and carry genes. Suggested image. Chromosome diagram with labeled DNA and genes. 5. Mitosis. Purpose of mitosis is growth, repair, and replacing damaged cells. Steps. 1. Cell grows, copies DNA, and increases organelles. 2. Chromosomes line up in the center. 3. Chromosomes move to opposite ends of the cell. 4. Cytoplasm divides, forming two identical cells. Suggested images. Cell cycle diagram showing stages of mitosis. Exam tip. Identify mitosis by visible chromosomes and dividing nuclei. 6. Binary fission in bacteria. Bacteria divide by binary fission, producing two genetically identical daughter cells. Division can happen as often as every 20 minutes under ideal conditions. Steps: 1. DNA and plasmids replicate. 2. Cell enlarges and DNA moves to poles. 3. Cytoplasm divides and new cell walls form. 4. Two daughter cells result. Example calculation. One bacterial cell divides every 30 minutes. How many cells after 2.5 hours? Convert hours to minutes. 2.5 hours times 60 equals 150 minutes. Number of divisions equals 150 divided by 30 equals 5. Number of cells equals 2 to the power of 5 equals 32. Suggested images. Diagram of bacterial binary fission. 7. Culturing microorganisms. Bacteria grow on agar plates or in nutrient broth. School cultures should not exceed 25 degrees Celsius to avoid pathogens. To keep cultures uncontaminated, sterilize petri dishes and inoculating loops. Tape lids lightly. Store petri dishes upside down. Testing antibiotics and antiseptics. 1. Soak paper discs in antibiotics or solutions. 2. Place discs on bacteria-covered agar. 3. Measure inhibition zones where no bacteria grow. Formula for area. Area equals pi times radius squared. Suggested images: agar plate showing inhibition zones with labeled discs. 8. Stem cells. Stem cells are unspecialized cells that can make more stem cells or differentiate into specific cell types. Adult stem cells, found in bone marrow, can produce certain cells, like blood cells, used in treatments such as for sickle cell anemia. Embryonic stem cells, found in early embryos, can become any cell type. Potential medical uses include replacing nerve cells for spinal injuries and insulin-producing cells for diabetes. Therapeutic cloning produces cells genetically identical to the patient, avoiding rejection. Risks and ethics. Lab-grown stem cells could carry viruses. Embryos are destroyed during research, creating ethical debates. Plant stem cells, found in meristems, can produce clones of whole plants quickly. Useful for crops and protecting rare species. Suggested images: stem cell differentiation diagram, plant meristem diagram, therapeutic cloning illustration. Exam-style questions and answers. Q1. Name the subcellular structure containing chromosomes. A1. Nucleus. Q2. How many copies of each chromosome are in a female human cell? A2. 2, one from each parent. Q3. Name the process for a body cell dividing into identical cells. A3. Mitosis. Q4. What is a specialized cell? A4. A cell adapted for a specific function. Q5. What is the unspecialized cell in an organism? A 5. Stem cell. Q6. A bacterial cell divides every 48 minutes. How many cells after 4 hours?
4 hours times 60 equals 240 minutes.
Number of divisions equals 240 divided by 48 equals 5. Number of cells equals 2 to the power of 5 equals 32. A 6. 32 cells. Q7. Conditions for fast bacterial growth. A 7. Warm temperature or plenty of nutrients. Q8. How to avoid contamination in bacterial culture. A. 8. Sterilize Petri dishes and loops, tape lids lightly, store upside down. Q9. Which antibiotic is most effective? A 9. The one with the largest inhibition zone. Q10. How can stem cells treat Alzheimer's disease? A 10. Replace lost nerve cells to restore brain function. Q11. Where are plant stem cells found? 11. In meristems.